high regulated levels of transcription in vivo require transcriptional regulatory proteins, the mediator complex, and nucleosome modifying enzymes, in addition to the above described general transcription factors. So these are the requirements for a highly speedy transcription. One reason for these additional requirements is that the DNA template in vivo is packaged into chromatin. This condition complicates binding of the RNA polymerase to the promoter and its associated factors. Transcriptional regulatory proteins called activators help recruit polymerase to the promoter, stabilizing its binding there. So these activators actually help the polymerase to bind with the promoters, and once it bind with the promoter, these activators then stabilize its binding. This recruitment is mediated through interactions between DNA bound activators, chromatin modifying and remodeling factors, and parts of the transcription machinery. So all these are involved for this recruitment. One such interaction is with the mediator complex. Mediator is associated with the basic transcription machinery, most likely touching the CTD tail of the large polymerase subunit through one surface, while presenting other surfaces for interaction with DNA-bound activators. This explains the need for mediator to achieve significant transcription in vivo. So this mediator complex, it helps to bind with the CTD tail on one side and to the DNA sequences on the other side, DNA activators on the other side. So this is the whole machinery, and you can see this is mediator. This whole blue portion is mediator. So in this, all the pre-initiation complex, there is RNA polymerase, and then there are different transcription factors. This one is the mediator complex. So it is involved in the transcription. Despite this central role in transcriptional activation, deletion of individual subunits of mediator often leads to loss of expression of only a small subset of genes different from for each subunit. So this mediator has very important role. But even if some subunits are lost from this mediator, it will not result into stoppage of transcription. Rather, it will just a small set of genes will be lost. This result likely reflects the fact that different activators are believed to interact with different mediator subunits to bring polymerase to different genes. So different mediator complex, different activators, they are involved in transcription. Mediator also aids initiation by regulating the CTD kinase in TFIIH. The need for nucleosome modifiers and remodelers also differs at different promoters, or even at the same promoter under different circumstances. So the nucleosome modifiers and remodelers, they are also different. When and where required, these complexes are also typically recruited by the DNA-bound activators or sometimes by regulatory RNAs. The yeast and human mediator each include more than 20 subunits, of which 7 show significant sequence homology between the organisms. So 7 out of these 20 subunits are very similar in their amino acid composition. The mediator from both yeast and humans is organized into modules, each containing a subset of subunits. So there are different modules which are present in the mediator. These modules, which are called head, middle or arm, and the tail, they can be dissociated from one another under certain conditions in vivo. So during transcription they are attached, but under certain conditions they can be dissociated. So here you can see dissociated form of the mediator. This is the mediator, and this is the rest of the machinery consisting of RNA polymerase and other transcription factors. So in this mediator you can see this is head, this is middle module, and this one is the tail module. So this is a mediator. Crystal structure of the head module of yeast mediator reveals that it contains 7 subunits, that is Med17 SRB4, Med11, Med22 SRB6, Med6, Med8, Med18 SRB5, and Med20 SRB2. So these are the subunits of head of yeast mediator. It forms a 3-domain structure that binds the transcription complex in such a way as to juxtapose TFIIH and the CTD tail of the RNA polymerase. So the arrangement is such that it juxtaposes these two. And why the position is such? Just to promote the phosphorylation of the CTD tail of polymerase by the TFIIH factor.